Okay, let's move on to the third part, AlphaFold. You perhaps have heard about this word AlphaFold, and the last section was organized for this protein structure prediction related part. So I'm not going to talk about too much details on AlphaFold, but more like the background and how it has solved this long-standing protein structure prediction problem, and also how to use it at the demo part. We learned that protein structures are very useful, but also that the painful process behind these experimental processes. So each experimental method got their own Nobel Prize, and many of the challenging protein structure determinations or determination of very important biomolecules also won Nobel Prize. There are about 20 Nobel Prizes that went to protein structure determination part. So that's a very important field, but also what means it's very challenging field. Alternative way of getting protein structures is modeling. If you can predict protein structures without doing these painful experiments, then it'll be a huge impact. And we also learned that proteins fold into unique structure when their sequence is given, and that follows the physical principles. So it was a dream for many physicists and chemists to know the folding principles so that we can predict structures. This has become a long-standing challenge in biology, also in physical chemistry field as well. And that dream actually happened very recently. This year, the Nobel Prize in Chemistry was awarded to AlphaFold and protein structure prediction part for one half. The other half went to protein design part for David Baker. And AlphaFold was invented by many scientists, and the main leader was Demis Hassabis, who is the CEO of DeepMind, and John Jumper, who led this AlphaFold project. So they made AlphaFold 2, which we think the solution to protein structure prediction part in 2020. So it's just 4 years since they made it and when they got Nobel Prize. So that means it was huge impact to whole biological field. In order to understand this protein structure prediction part or this area, we first need to understand a competition called CASP. CASP stands for Critical Assessment of Structure Prediction. And it's a kind of Olympics which happens every 2 years. So this year there was a CASP 16, which means the first CASP was happened 30 years ago in 1994. And the important and interesting part of this CASP competition is that it's a fair blind test of computational methods for protein structure prediction. So there's a group of people called assessors or organizers who mediate between experimentalists and predictors. Only they can access to all the information. So they first get the problem, which means protein structure, and input, which means protein sequence, and then give it to predictors. And predictors, of course, doesn't know what the protein structure look like. They could only get the protein sequence and then return back what they think the protein should fold into. And after having all these, assessors compare against the answers to the model structures predictors made, and then announce the evaluation results at the end of year. So this is how typically CASP goes. And giving you an example, what happened this year. In CASP 16, the first target was a T1201, which had 210 residues. And if you register here, you could only get this information, and that's it. And you can see that there's a heat stack here. We learned that it's not meaningful protein sequence. So you only need to model about 200 residues here. But you don't have any other information. So you have to put this sequence into your computer and using your own computational method, you have to build some kind of 3D structure looking like this in 2 weeks. And then this kind of targets are released for a whole 3 months. And they contains about 100 targets every CASP. And organize, collect every them, and then analyze by comparing to the answers, and announce at the end of year which method did the best performance against the others. This is how typically CASP works. This CASP has started with a lot of ambition, but then nothing much progressed since 1994 for about 15 years. This is a metric showing how accurate the protein structure is against their native structures. And on average, for 15 years, people were the scores of people were about 40 points. And 40 points is not a good score when the maximum score is on 100. And this is an example of 50-point score in GDT-TS. And you can see that they are roughly correct. They match about in fold level, but definitely they are not in family level. And this was the best what they could have done until 2010. And the story started to change since 2016. People discovered how to fold proteins not from their principles, but from history. Here, history means evolutional information of proteins. So you know that proteins are shared across different organisms, and that shows how that protein has been evolved through the history and simulated by nature. So if you collect the same protein from different organisms like this and build an MSA, you can collect which residues are conserved, shown in orange here. But also you can collect residue pairs which co-evolve. Co-evolve means when some part changes, the other part follows. And this is called coevolution information. And the people thought the origin of coevolution is the structure, 3D structure. They co-evolve because they have to stabilize when something is mutated or compensate that mutation. Imagine like this. When these red parts mutate into green, then no longer green was happy. So you have to co-evolve in order to make this protein survive. So people thought this 3D structure closeness, or what you call contacts, is the main origin of coevolution. And by analyzing this MSA, you can collect many contacts, residue pairs that are contacting to each other. And by using that information, you can build more accurate structures than before. But then there was still about 10% improvement in the score. And then suddenly 4 years after, everything was solved. And that was mediated by AI revolution. At the end of 2020, BBC announced that one of biology's biggest mysteries largely solved by AI. So they didn't say it was solved, but they said so largely solved, because at the time it was not very they were not very certain about this, but now we say it's solved by AI. And later on in 2021, both journals Nature and Science announced that this is the breakthrough what was made in 2020. And how could you say this is solved? If you look at the absolute score in CAS, you can see that there's a jump in AlphaFold 1 and another jump in AlphaFold 2, which made their average score of 90. And score 90 is solved because proteins are dynamic. And you don't have to make 100. 90 is about a great number. You can get the protein structures correctly. And another reason why we say this problem is solved is because not only because it's accurate, but also because it generally works. People usually did a very nice job on getting protein structures when there's a good template, which means a very close homolog structure deposited in PDB. But even if this case, when there's no close template, and when close to the template varies a lot from the experimental structure, AlphaFold could get much, much better structure than just copying the template, which means AlphaFold works generally for most of the proteins. So it's accurate and generally applicable, so we say it's solved. And how this was, how come this happened? The logic is same. It's still using not the principle, but the history of evolution. This is a figure I brought from AlphaFold 2 paper. And the major part in this algorithm is actually input here. Sorry. MSA. There are other minor inputs, but you can just ignore them. The 99% of information comes from this MSA. And previously, people tried to build protein structure by statistically analyzing this MSA and extracting coevolution patterns. But this pattern analysis could be done much, much better using AI. So that's a major difference. And in AlphaFold 2, this following AI part analyze which part, how to extract correct context and distance information from MSA to build this 3D structure. This was how this AI plus coevolution information enabled the protein structure prediction problem solve. And this is not the end of story. Because proteins share the same logic, this could be also made for protein-protein complex prediction or protein-peptide complex prediction. By slightly changing the AlphaFold 2, they could build a new AI for multimodal prediction. And with this, you can build a very nice 3D structure of complex structures. And the result was much, much better than other existing protein complex prediction tools. And same story for protein peptide prediction. It could achieve the state-of-art prediction results compared to previously published methods. So AlphaFold 2 was not just solving protein structure prediction problem, but also gave a huge impact to any kind of amino acid related prediction problems. And not just that, but also they build huge number of model structures for known proteins whose sequences are known, but structures are unknown. So you can access to AlphaFold DB here, and then get whatever structure you want, for which you don't have structures. And at the demo part, I'll show you how to use this. And these days, because protein structure prediction field has been solved, people are trying to move beyond that. For example, Google DeepMind published recently a new model named AlphaFold 3, new AI, that not only models proteins, but also nucleic acids, ligands, metals, and post-transcriptional modifications, and interaction between all of this in atomic level. So here's the example of what AlphaFold 3 does. So this is becoming more accurate and more general structure prediction tool for biomolecules. So from here, we'll get a short demo on how to use PDB website and how to extract your own model structure from AlphaFold DB when you don't have anything in PDB.